Good morning, everyone. My name is Pierre Domel, co-founder and CEO of InGel Therapeutics. Very excited to share with you some of the recent development that we've been working on at InGel. And I would like to thank, of course, the Alliance for Regenerative Medicine for giving us this opportunity today to share with you some of this work. So InGel focuses on ophthalmic diseases. And our mission is to bring back light to patients through regenerative medicine. More specifically, patients that are losing their sight due to retinal degenerative diseases. And to do so, we use two of our main innovations. We use those two in synergy with each other. So we work on the therapeutics itself. We have created a regenerative cell therapy platform. So we created a protocol to expand and isolate human retinal neurons in high purity and high viability. From primary donor cells, we're now able to get 95% pure rod photoreceptors or cone photoreceptors or even retinal ganglion cells. But we also thought about how can we work on the delivery mechanism of those cells? How can we make them more viable, more efficacious in vivo? So with the tissue engineering approach, we created a biomimetic delivery scaffold to encapsulate these cells and enable a better efficacy in vivo within the eye. So this hydrogel is tunable for intravitreal and subretinal delivery, but it can also be used for other applications within the CNS. So we want to use first these two innovations to target our beachhead indication, which is retinitis pigmentosa. RP is a group of inherited retinal disease that causes progressive vision loss, and it has a very high and huge unmet need as of today. I'm showing you here a few pictures and a few schematics of the retina just to simplify what's happening in RP in patients. At the early stage, RP starts with one out of more than 150 different mutations, primarily in rod cells, or just not in cones at least. So the green cell that you see over there. This cell dies. The mutation caused the rod cells to die, and therefore patients, as you see on the picture, they lose their peripheral or side vision. But next step is actually even more critical. The death of the rods leads to the loss of the cone photoreceptor cells. And these cones are critical for your vision. They're responsible for your color vision, visual acuity, and central vision. Basically, you use your cones to watch the screen, to read a book, watch TV, work on your computer. So they're very, very important. And due to a bystander effect, without any mutation, these cones die in retinitis pigmentosa. So you can see that for patients, they start to have a very narrow tunnel vision. And then when mid and late stage happens, they lose the color vision, and then it's complete blindness because the cones are entirely dead and they are basically blind. So at InGel, we're focusing on a mid-stage approach, a gene-agnostic mid-stage approach to treat retinitis pigmentosa, targeting patients in this mid-stage that do not have rods anymore, but have cones that are in the process of dying or not functional anymore. And to understand this, we looked at what are the 4 primary drivers that are leading to this death of cones and their loss of function. You have a metabolic deficiency. Basically, these cones are starving to death. They do not make ATP, they do not make glycolysis, and they do not have energy to survive anymore. Second, very well known, you have a lot of oxidative stress in the retina and inflammation. The high amount of ROS species and also all the lipid peroxidation ruptures the cells and kills them. And then with those two, the cones, they start to lose their primary functions. The first one being performing phototransduction, so converting the light into an electrical signal. And the second one is the synaptic connection loss. Basically, the cones lose their ability to transfer that information to the rest of the retina, the inner retina, and then later on to the visual cortex. So to combat those 4 primary drivers, we created our first asset, IGT-001. It is an intravitreal stem cell matrix to restore photoreceptor functions. If you remember the 2 innovations that I mentioned, we basically bring 2 together, our rod cells, so rod photoreceptors, that are encapsulated within one formulation of our hydrogel within the syringe. But not only we encapsulate the rods, we also encapsulate all the vesicles, microvesicles, exosomes, proteins, and enzymes that our rods are secreting. And those proteins and enzymes are critical for cone rescue. So it is an injectable technology. So we put it in a normal syringe and perform a simple intravitreal injection of this cell matrix. And the technology then works in 2 steps. The first step is that we've designed this hydrogel to adhere onto the retina, onto the first layer, which is called the internal limiting membrane. It doesn't float within the eye. And the second step is that you can think about this as a biofactory. Our rods will secrete all these factors that I mentioned before in order to protect the cones. This factor will be released from the rods to the gel within the retina, would be transported through the retina, targeting the cones in order to rescue and repair these cones. So to be very clear here, our stem cells are not being used for cell transplantation. Here, for our first asset, they're being used as a biofactory to provide trophic factors for the cones and to rescue them. With all our preclinical studies, we've been able to show that IGT-001 can perform a dual action. First, we can do what we call neuroprotection, rescuing existing cones. That's what you see on the bottom at the mid-stage. Those are cones that are in the process of dying. Some of them are still functional, but we're able to give them enough trophic factors to make them extremely viable. But what we did is that we actually took IGT-001 to the next step. And we went a little bit later. And now we're able to show, and I'll show you some results, that we're able to do neuronal repair. Basically, those are cones that are dormant cones. They do not have outer segments anymore. The cell body is there. However, they do not perform. They do not function. And we've been able to show that with IGT-001, we're able to give them the factors they need to bring them back to a viable state so that they actually start functioning again. All right, so I want to show you, both cone rescue and neuronal repair in terms of a couple of, preclinical studies that we've performed. Here, first focusing on functional rescue, we've done a preclinical study on one animal model, which is called the Rho knockout or rhodopsin knockout. This is a mutation that happens in about 20 to 25% of patients with RP. And here we're looking first at visual behavior, basically the behavioral vision of these mice. We can measure both contrast sensitivity and visual acuity. And what you see in these graphs is that after injection at 21 days old mice, we actually are able to significantly improve both contrast sensitivity and visual acuity with these mice by injecting IGT-001 in their vitreous. Of note is that if you look, for example, at contrast sensitivity, you can see that we designed this hydrogel to degrade in about 45 days in mice. And you can see for the period of 45 days, the contrast sensitivity is just not dropping. We're able to halt the degeneration of these mice compared to the control or the sham group in gray during the time that the hydrogel stays within the eye with our cells. So this is for visual behavior or behavioral vision, but we also wanted to check for functional vision. And for that, we perform on the same mice electroretinography, ERG, focusing on the photopeak B wave, which is basically looking at cones function. And we actually were able to see almost exactly the same trend, being able to halt almost entirely the degeneration of ERG during the time that the hydrogel stays within the eye. And this was confirmed with our anatomical results that you see on the right. In red, those are the cones. And in IGT-001 group, we were able to see a higher number of cones, morphologically accurate and viable cones with outer segments that are performing their function of phototransduction and transmitting this signal to the rest of the retina. So that is for cone rescue, but I also wanted to share, and that's very recent data over the past few months, the neuronal repair part. For that, we took another animal model, a very aggressive one. It's called the P23H mouse model. Briefly, basically, this mouse model is entirely blind at 4 weeks old. And we decided to inject at 4 weeks old when we knew, there is no behavioral vision, no functional vision, nothing anymore. So what you see on this graph right here is at 4 weeks old, the ERG is almost zero. It's just noise. There is nothing there. But what we were able to show is that even 1 week after injection, we were able to rescue almost half of the ERG B wave in these mice with IGT-001, and very similarly with just our cells alone without the hydrogel. We actually made that study very fast because we wanted to see within the retina with staining and with anatomical readout what is happening to these cones. So what you see on the right are these anatomical staining, and not only in the IGT-001 group, we were able to see cones with their outer segments being morphologically viable and functional, but also that their synaptic connection to the rest of the retina was restored and rescued, meaning that we were able to, at the same time, restore their phototransduction mechanism and their synaptic connection to bipolar cells and other cell types within the retina. So just going back to the 4 primary drivers that I mentioned at the beginning, with these preclinical studies and other preclinical studies we perform, we're able to show that IGT-001 restores the homeostasis of the retina, targeting multiple pathways in cones. And to be a little bit more precise, we're targeting 2 major pathways. The first one is glycolysis. We have one target, which is RDCVF, rod-derived cone viability factors, which enables glucose to be uptaken by the cones. But the second target is the lactate pathway, very critical for us, enabling basically the transformation or the production of lactate, transformation from glucose to lactate, and making ATP and energy for these cones. And the second pathway we're targeting is oxidative stress. The first target is peroxidation. So we basically protect the cells from the lipid peroxidation and prevent the cell membrane damage. And the second target is a well-known pathway, oxidative stress pathway within the retina. It's called the NRF2 pathway, where we at the same time can produce NRF2 and activate NRF2 in order to secrete antioxidants within the retina. Right. So just a little bit briefly about the development plan of IGT-001. We're about 18 to 24 months away from the clinic right now. We're currently raising in order for us to reach IND by the end of 2025, focusing on manufacturing, of course, and regulatory pathway with our NHP and tox study. And the team behind InGel is actually very passionate about making this a reality. Myself and Patrick, our Chief Business Officer, are here at the meeting, so if you have any questions and you want to chat with us, please feel free to come to us after the talk, of course. And we have amazing scientific advisors from Harvard Medical School and a stellar business and clinical advisory board who are steering us the right way to make IGT-001 a reality for patients one day. So with all of this, thank you so much for listening. I'm more than happy to answer any questions you might have, and I hope you can join us in our journey to bring back light to our patients. Thank you.
What's the cell you deliver nutrient or growth factor to our proteins in the retina? Is the epicellular cells or the cone cell itself?
So can you say again?
Is a retina pigmental epicellular cells for the growth of the cone or directly to the cone cells?
So that's a very good question. The factors, we've been able to show that they go to the cones, these factors, to target glycolysis within cones, oxidative stress, but those factors also go to the rest of the cells in the retina. Some of them are being uptaken by Muller glia, some of them by retinal pigment epithelial cells. I showed at one point the lactate pathway. It's actually an interplay between cones, rods, Muller glia, and RPE. So we're providing factors for the entire retina, of course, but we target at the end cone rescue.